Thank you, Volker. Good morning, everyone. Thank you for coming for this talk. For the full disclosure, this is invitation talk from GenScript. GenScript, we collaborate and partner a lot in the recent years on many different projects. So predominantly on large-scale expressions of antibodies and proteins, as well as the different synthesis technologies that they have. But the talk is about Oceanics and the company that what we're working on, and that is brain shuttles and delivery of therapeutics to the brain. We're predominantly focusing on antibodies at the moment, but also have lots of experience with delivery of different therapeutic payloads, such as antibodies, peptides, enzymes, as well as oligonucleotides. So first of all, what I'd like to introduce you is VNOS. These are single domain antibodies that are naturally found in sharks. We are mostly working with the business part of the antibody, which is the variable domain, which is called VNO, hence the VNO antibody that we're using. There's no really any homology between single domain antibodies from sharks and camelids, and they're obviously very different to conventional antibodies that are built of heavy and light chain. One interesting thing that I would like to mention here is that the single domain antibodies in camelids as well as in cartilaginous fish, they exist alongside conventional antibodies, and therefore you might think that there's a different class of targets that those antibodies are probably targeting. So there's some form of added function of having single domain antibodies in those animals. And when you look at the crystal structure from the top view, and on the left-hand side you have Fab, VHH, and you have VNO on the right, you can see the footprint of the antibody is very different. When you look on the side view, additional thing that you see that in VNOs you have those CDR3 loops that are protruding and are able to bind to concave structures in the proteins. So looking simply on the structure, you can realize, or you can assume that those antibodies will be able to bind to different epitopes, and that's actually is the case, and that was confirmed in many of different publications that show that VNOs bind predominantly to structural epitopes, and they don't share the same epitopes with conventional antibodies. So that's where we think is the antibody can be used for those special tasks, somewhere that you cannot either target with IgGs, or somewhere where you like to have a different function, different biology. So I'm talking about the blood-brain barrier. We were working on different projects, but in Oceanics we pretty much defined our sort of aims to develop the best-in-class brain shuttles. So blood-brain barrier is a natural barrier that separates blood from the CNS, from the brain. It's built of tightly packed endothelial cells, parasites, and astrocytes. And it's great to have that because the barrier prevents large molecules and toxins and bacteria and viruses freely entering that critical organ. The drawback of that is that many therapeutics, such as antibodies, peptides, oligos, cannot enter the brain, and there's very little efficacy of those drugs in the brain. Over the years, many different methods of tackling blood-brain barrier has been developed. The most sort of elegant, I think, and the most clinically advanced method is based on receptor-mediated transcytosis, when you develop antibodies against receptors that are facing the luminal site of the blood-brain barrier. And those receptors normally transport different natural ligand across the blood-brain barrier to the brain. One of those is transferrin receptor 1, which is most commonly used in the field and advanced in the clinic, which transport transferrin delivering ion to the brain. So you can identify antibodies against transferrin receptor 1 as we did, that allows them to be transported to the brain and delivering fused payloads to the brain. That's not as easy as it possibly sounds. There's a particular interplay of binding epitopes and binding kinetics to the receptor that allows successful transcytosis of those antibodies. So the antibody has to initially bind to the receptor. So in our case, we identified that the association rate is the most critical parameter that dictates a successful brain penetration. And then subsequently, during the transcytosis, the molecule has to be released. In our case, the antibody binds to the structural epitope, and we believe that during the transport and the structural changes of the receptor, the antibody can be released on the parenchymal side. So that's how we develop we believe it's clinic-ready brain shuttle that we call TXP1. And just give you a few slides on the biology of the behavior of the antibody. So this brain shuttle TXP1 has very nice binding and identical binding to human transferrin receptor as well as monkey cyanotransferrin receptor. Doesn't cross-react with mouse or rat transferrin receptor. As I mentioned before, one of the most important parameters that we pay attention to is the association rate. And in that case, association rate between human and monkey transferrin receptor 1 is practically identical. And that's why we believe that the molecule, after testing in non-human primates, can be successfully translated to the clinic. The antibody binds also to the model blood-brain barrier cell line that is used in the field, and after binding, it does internalize and colocalize with the natural ligand transferrin. Importantly, one of the characteristics of the brain shuttles based on transferrin receptor 1 antibodies is that the antibody cannot have competition with the natural ligands because you don't want to affect the ion transport to the brain, which can be something that you don't really want. So we're testing that extensively during the screening process, and one of the methods that we looked at is the internalization in HCMECD3 cells of TXP1 on its own or in the excessive presence of transferrin. And you can see on the right-hand side the internalization of TXP1 without or with transferrin is unaffected, unlike the other antibody that we use as a control that we know that competes with transferrin for the TFR1 binding and internalization. We've done quite extensive studies on that. That was the internalization study. We've done in vitro sort of study that's based on octet, where we also see there's no competition with natural ligands such as transferrin or ferritin. Another important aspect that we found that allows you to successfully use the antibodies as brain shuttle is the ability of that antibodies to recognize endogenously express TFR1. We're predominantly working in the earlier stages with the recombinant TFR1, but a lot of antibodies that we developed through that process do not recognize endogenously TFR1. And we like to test it by immunohistochemistry. And TXP1, you can see, binds really nicely to human and monkey sections from frontal cortex. And when you look a little bit closer by immunofluorescence, you can see that TXP1 binds to the structures such as small and large capillaries on the left. On the right, you can see nice binding to TFR1 expressing neurons such as Purkinje cells in cerebellum. So now, when we go to the in vivo testing, what do we see? So the molecule of choice that we work with, you can have the cartoon on the top left. It's two vinos attached to human FC domain. That's the sort of our molecule that we do a lot of sort of initial studies. As when you inject that to monkeys, approximately at 5 mg/kg, then collect the brain after perfusion and measure the concentration in different regions of the brain, you can see very nice levels in pretty much every organ of the brain of TXP1 in comparison to isotype control antibody that we use. So in terms of fold difference, depending on the dose, with 5.4 mg per kg dosing, we have over 35-fold increase in comparison to isotype control in pretty much every region of the brain that we've tested. Another unique feature of the antibodies that we're working on is specificity to the brain-expressed TFR1. We've developed, over the previous 5 or 7 years, we developed a mouse brain shuttle, which has very much the same unique specificity to the brain, not only able to penetrate the brain, but doesn't bind to different organs that express TFR1, such as liver. Using those methods that we apply in the screening process, we identify TXP1 that seems to have very similar characteristics, and it's brain-specific. The molecule also can be measured in CSF, which is very important. It's an indirect measure of penetration across the blood-brain barrier, but also gives us a nice opportunity to measure molecule in potential future clinical trials in CSF sampling. Now, about a little bit of the PK data that we have for TXP1. We would like to have more data, but that's what we have so far. So looking at the brain concentration on the left-hand side, we tested animals dosed at 72 nanomole per kg. This is 5 mg per kg. And looking in the perfused brain day 1 and day 4, and you can see practically there's unchanged level of TXP1 in the brain over that 4-day period. When you look at the plasma levels, the molecules undergoes normal elimination. So when you look at the elimination rates or elimination slopes in the brain and plasma, you can see that molecule is practically unchanged in the brain, and in plasma it's undergoing normal elimination. That is a strong evidence that, unlike with small molecules, this is active transport of the molecule across the blood-brain barrier that is partially independent, at least in this dose range, from the plasma levels. So when you look at the brain to plasma level over a period of time, you can see that it's increasing. Now, the direct evidence I would like to show you for the penetration to the brain parenchyma. Lots of molecules that have been developed as brain shuttles have ability to recognize TFR1 or the blood-brain barrier, but they also stop there. They being not effectively transporting across the blood-brain barrier to brain parenchyma. So this is immunohistochemistry staining from the monkeys injected with either isotype control or TXP1, and you can see very different picture in the hindbrain. When you zoom in, you can see very nice capillary staining through which the molecules are reaching parenchyma, and you can see also accumulation in neurons, such as in cerebellum, in Purkinje cells, but also very nice accumulation in different brainstem regions. So I mentioned, like, previous studies show that previous groups published that some TFR1 antibodies are binding to the receptor on the blood-brain barrier, but they also potentially deplete endogenously expressed TFR1 from the blood-brain barrier. So we look at it as a potential liability of antibodies, and we look through different methods. That's just quite a nice method by using immunofluorescence, where we look at TFR1 and endogenous TFR1 in the sections contained with lectin. So we were basically quantifying specifically TFR1 in the capillaries. And when you quantify that, you don't see any drop in endogenous TFR1 in animals treated with TXP1 in comparison to isotype control antibody. We also didn't see any reticulocyte depletion that expressed TFR1. That's mostly predominantly by the fact that we're using FC domain with LALA-PG, so it's effector function dead. So even if the molecule will bind to those, we won't see depletion. Now, coming to the application of these antibodies or these brain shuttles. So this is just the case I would like to show you, our fusion to anti-HER2 antibody trastuzumab. We are interested in different modalities and different antibodies, such as agonist TrkB antibody for Parkinson's disease, TREM2 or CD20 for multiple sclerosis, as well as I mentioned, oligos for many different indications. But here I will talk a little bit more about TXP1 HER2 fusion for neuro-oncology. And why is it interesting? Why we're working with that is because HER2 is overexpressed on multiple cancer cells. Approximately 30% of lung and breast cancer patients are HER2 positive, and out of those, approximately 50% of those patients will develop brain metastasis during the course of the disease. There's very limited treatment options for brain metastasis in those patients, mainly surgical resections and radiotherapy. And then there's a very poor prognosis. So there's only approximately less than a year median overall survival for patients that have been diagnosed with brain metastasis. And the population size is huge. Just for the US and Europe, there's over 160,000 patients that can suffering from such from brain metastasis every year. So our idea is pretty simple and straightforward, combining trastuzumab, that is efficacious drug systemically but doesn't penetrate blood-brain barrier, with our brain shuttle that improves delivery of the molecule to the brain. So upon delivery, trastuzumab mode of action is by reducing the proliferation rate of the tumor cells, as well as able to recruit CNS and infiltrating immune cells. So now going to the biology of those molecules. On the top left side, you have the cartoon of how we like to sort of work with the antibody delivery. We prepare there's a bispecific, bivalent molecule for TFR1 with N-terminal fusion. Other formats are, of course, possible, and we're working with that. Some are better, some are worse, but this is one of those one of those formats that we like to start with. And in terms of HER2, you can see that fusion of TXP1 to N-terminal end of heavy chain doesn't affect HER2 binding, as you can see on that ELISA graph. We also have SPR to confirm those observations. We also have very nice binding, retained binding to transferrin receptor 1, and approximately 10 nanomolar affinity. And again, the same for human and the same for monkey. We have very nice binding, retained binding to the model blood-brain barrier cell line, and we have also internalization as we would have just with TXP1. So when we inject that molecule to monkeys at the same nanomolar dose as before, 72 nanomolar per kg, in this case it's approximately 10 mg per kg because of the molecular weight, and we look at the biodistribution in the brain, we're reaching pretty much the same concentration in the brain as we had before with TXP1 fused just to the Fc domain. As you can see, we have 8 nanomolar concentration in all different regions of the brain, including cortex, striatum, thalamus, hippocampus, midbrain, and cerebellum. And again, we have a very high level of brain specificity with no accumulation in other major organs such as liver, spleen, heart, muscle, and kidney. So this is just the average data on the brain, approximately 8 nanomolar concentration. We have nice exposure in CSF. And then when you look at the plasma levels, and something that should be appreciated is the fact that the molecule on its own, HER2 antibody alone, and TXP1 fusion to HER2 practically has the same profile in that really short period of time. But basically, elimination rate in that elimination rate and the slope of elimination for HER2 antibody is the same as for TXP1 HER2, so there's no major disposition of that antibody to any other organs. So that confirms our biodistribution, that shows the specificity to the brain. And also that can be assured by when you calculate volume of distribution upon infusion, is practically the same for HER2 and TXP1 HER2. So overall, what we think we developed is an industry-leading brain shuttle, has very high brain specificity, high parenchymal penetration with delivery of different therapeutic payloads, such as antibodies with improvement of over 30-fold improvement. We have superior PK profile because of that brain specificity, so we have prolonged exposure in blood, but also in the brain. So it's not a transient process. The molecule keeps going to the brain over a period of time. The molecule is cross-species reactive, so can be directly translated from monkeys to humans. We fully deimmunize the VNR scaffold, confirm developability, and optimize formatting for antibody and different modalities. And we also have a brain shuttle that we use for proof of concept studies in mice that mimics lots of those characteristics that we have for TXP1. So this is just a slide, just compares different technologies and approaches used by different companies such as Roche, JCR, and Denali. So everyone looks like using different methods for delivery of the therapeutic payloads to the brain. What differentiates our technology is not only the fact that we can deliver the molecule across the blood-brain barrier, but we have very high specificity to the brain. So therefore, what comes next is sustained brain exposure that we can extend for a number of days. We have long plasma half-life. The molecules, again, is cross-reactive. The formatting of VNRs and putting them on in different formats and using it for delivery of different modalities is relatively simple. And yeah. So with that, I would like to thank all the people from Oceanics who work on that project, as well as our long-standing academic collaborators. And again, I would like to thank GeneScript for giving us opportunity to talk to you and inviting us to give this talk. And I suppose to mention that we're using GeneScript for that turbo expression system for a lot of different protein expression and antibodies. And they became our sort of CRO of choice to go to. And they will be happy probably to talk to you about the ability. So thank you.
Thanks very much, Pawel. Some of you might have asked or asking yourself, where was AI in this talk? This is,
you can explain.
As Pawel says, this is a GeneScript sponsored talk, and the organizers slotted that in this session. So I believe it's the only talk that has no relation to AI in this session. But it was nevertheless very interesting. Thanks very much, Pawel. We have some time for questions. Are there any questions from the audience?
Just two questions. The first one is, have you validated your target outside of steady state conditions, for example, under anemia of inflammation? And the second one, what's the difference on the receptors that gives you that specificity to the brain?
So I can start with the second. So there are different post-translation modifications to the receptor, and it looks like our VNR antibody is able to recognize them differently. So that's what's probably happening. In terms of different conditions, or like steady state in wild-type animals, we also have, not with TXP1, but with the brain shuttle for the mouse, we tested that brain shuttle in conjunctions with different therapeutic payloads in different animal models. So in the Parkinson's disease model, we tested it successfully, as well as Alzheimer's. We tested it with delivery of enzyme in Hural syndrome model with iduronidase knockdown. In spinal cord injury as well. So.
Any other questions?
Just kind of follow on from that question, you mentioned that you're targeting specifically the receptor in the brain expression, and you're saying it's potentially because of distinct post-translational modifications. Is that something that you were specifically looking for, like, either during planning strategies or something like that, or was it just you selected the lead candidates that were only binding to the brain-expressed version of the receptor?
No. So molecule was selected based on binding to recombinant protein, but during the screening process, we included a number of steps that allow us to at least have high hope for the specificity to the brain, using predominantly immunohistochemistry and looking at binding to different tissues from different sections from different organs.
Great, thank you.
Okay, I was wondering as well, I think in the table you showed that the retention in the brain of the other molecules is different. Why is that? Because they also TFR1 binding molecules. Why is the TXP1 retained and the others are not?
Well, it's due to the fact that others are being eliminated quickly by disposition to liver mostly, which express high level of TFR1. So the other sort of competing technologies don't differentiate between different post-translational modification on TFR1 are being quickly eliminated from plasma. And as soon as you go below certain level in plasma, there's practically no penetration to the brain. You have to retain certain level in plasma that allows you to keep active transport.
Okay, so you're losing the systemic exposure to the blood-brain barrier, basically. It's not there anymore to be shuttled across. Yeah, okay. And how many sort of existing antibodies you think there are that could be hijacked with that? I mean, you showed Herceptin as an example, but how many other existing monoclonals do you think there are that could be just now shuttled across and...
Yeah, well, there's no limit, right? Anything you can format with brain shuttle and deliver. You have to make just the case for clinical use of those antibodies. But we've tested in mice, we've tested with different antibodies like EGFR, PD-L1, CD20, and some more, I think 5 or 6 or 7. And all of them, there's no difference how those antibodies penetrating the brain. So.
Okay, thank you. Any more questions? Oh.
Hi, Pawel. Thank you very much for the presentation. Linda here, so working with the GenScript, and maybe I can share a bit of comment on why we wanted Pawel to present today. So obviously we understand that the slot was for AI ML, and I will say a bit how Turbojo platform can help that, but we thought that Pawel has a very cool and very necessary technology, and we did want him to present today. We could tell that many of you were interested, so definitely come and talk more with him. He will be in the conference room and at times at our booth also, if you have more questions. But regarding GenScript, Turbojo platform that Pawel is mainly using for large scale, regarding AI, machine learning, what we are doing now is that actually many people are doing or need actually high throughput production of antibodies at the beginning, just microgram scale. So what we do is that for AI companies who come to us with even thousands of different antibodies that they want to get expressed, basically today with the Turbojo platform, we can do that as fast as 5 business days, which is the fastest recombinant antibody production on the market. There are a few Nature articles mentioning that. It can be full-length antibodies, it can be VHHs, it can be bispecifics. So yeah, definitely please come talk to us regarding how we can support a bit more AI and machine learning. And please come talk to Pawel because yeah, what he just presented here is very much necessary.
Right. Thank you, Linda.
Yeah. Thank you for explaining.